All right. So thanks, Rock. Rock is putting to kind of organizing everything for the, along with Dorian, who just stepped out of the door, who's helping to organize everything for the lecture, all the mechanical stuff, having all the lectures recorded and everything like that. So okay, so this begins our 2017 summers course, and I can't even remember how long we've been doing this, at least since 2012 or so. So and hopefully it keeps on getting better every year, and thankfully we keep on getting the agreement from, I know this is actually way too small, but this schedule is actually well, I sent everyone a schedule, and if you don't have a schedule, let me know, and if you're not on the fMRI course listserv, let me know that as well, because you can actually sign up yourself. I sent out an email showing you where to go, showing everyone where to go to sign up. But if not, you can just email me and I'll try to put you on. But either way, the course is listed. It's on the website as well, on the fMRI, Functional MRI Facility website, which is this one right here. And so it might be updated, it might not be, but well, it will be in terms of some places we don't quite have a room yet. And actually, we were supposed to have this lecture in Building 40, but apparently Bill Gates is in town, and he's apparently doing something in Building 40. So that's why I was moved over to here. But anyway, just like to thank first of all of our lecturers. We have a huge amount of imaging talent here at the NIH, and not only for fMRI but also for multimodal applications, and also neuromodulation. So there's a number of people here. I won't go into the details because it's almost impossible to read anyway, but a lot of good lectures lined up. 41 lectures across the summer, 3 lectures a week almost every week. At least 2 weeks we'll have 4 lectures a week because we had to squeeze things in. We don't have one lecture during the OHBM. We couldn't find anyone to give a lecture then. I know there's 2 other lectures during OHBM, but hopefully there won't be too many. Those are the people, obviously, they're not going, but hopefully the people that aren't going also will get something out of those lectures. So, and not only does a course go over multimodal imaging, it goes over the basics of MRI, it goes over MRI applications, it goes over a lot of fMRI processing. So enough to sort of hopefully give you a sense of what's going on at the NIH, and also a sense of the basics and the cutting edge of what's possible. And so I usually like to kick off this lecture, though, with kind of a general overview of a really light sort of overview of the history of neuroimaging, and a little bit more detail, because I'm more familiar with it, is the history of functional MRI. And okay, so I'll just jump into that right now. So just starting out with a brief history of neuroimaging. And I was actually trying to, when I was researching this a little bit, I was trying to find, okay, so you can go back very, very far with that. And you can go much further back than the 1850s, but most people bring up, when they talk about brain mapping, lesion-based mapping. And that is the idea where someone hits their head, causes some brain damage, and they see how they behave a little differently based on that. And then they say that part of the brain or the head is very important to that behavior that was lost. And the first, I think, lesion-based mapping case that's also the most popular and the least conclusive is that of Phineas Gage. So in 1848, this guy was working on a railroad, and he was foreman, and people were, I think he was working on these tamponing rods that were pushing down something for explosives, and the explosive went out prematurely. This rod kind of went right through his sort of mouth and his head, and surprisingly to everyone, it didn't kill him, and even more surprisingly, he sort of got up after that, and he's like, oh, it must have gotten hurt a little bit. So they were surprised. So the reason why it's the least clear is because everyone was sort of shocked as to, this thing went right through his brain, and he wasn't that affected. And also, there's still debate even today of how much he was affected. Obviously, and so this part of the brain, while it's not important to basic functions, it's important to personality and decision making. Later on in his life, he ended up being a lot more impulsive, a lot less good at making rational decisions. His life was a lot rougher after that because his personality changed. But it was sort of unclear, and so that's why it's still interesting and debated. A little bit more clear studies where, for instance, Paul Broca in 1861, he had a patient who could only say the word tan and clearly had a lesion in this area that's now known as Broca's area for forming words. And then you have Wernicke, who also has an area right here that's named after him because he had a patient who couldn't understand or produce meaningful speech but could articulate words. So that area is involved with that sort of deficit. Okay, so lesion mapping only went so far, and to some degree it's still being done to some degree today. There's still some benefit to it because you can get a definite sense of causality from it. I mean, if you lesion an area and you have a behavioral deficit, you know that that area was important. Sometimes in functional imaging, if you see an area active, you don't necessarily know if it's causal for the behavior. So it's good in that regard, but it's still obviously a little bit more crude. So anatomic imaging. So just a quick overview. Even though X-rays aren't used today, it's still always useful to take a look at the history of that. Turns out in 1895, a big thing being studied was Crookes tubes, where basically it was a vacuum tube with an anode and a cathode, and it would cause, if it was the right material on the cathode, on the anode, electrons would be accelerated. As they're breaking, they would emit rays, and certain wavelength of ray called the X-ray, a very high energy ray that was discovered by Röntgen because he thought he had the tube covered up completely, but on the opposite side of the room there was some fluorescent material that was actually glowing even though he thought he had everything covered up. So in other words, it was a very high energy ray that was able to penetrate everything and then cause that fluorescent material to glow. So that's how he discovered X-rays. He since went on to further develop that. And these are some of the advantages or some uses of X-rays or that energy of radiation. Okay, so with X-rays, people were trying to get more and more interesting information about, for instance, brain physiology or blood vessels, or even to enhance contrast. So one thing that they did in the early 1900s, which seemed like torture, in order to get better contrast between the tissue and open air, they figured the CSF, which had the same density as the tissue, they figured they would just drain the CSF, and then they could have nice contrast. I mean, people were pretty crazy, I guess, or committed or something. So that only hung around for a while until they had better ways of enhancing contrast with X-rays. What's also interesting, though, this guy, the first Antonio Moniz, who created the first arteriogram using sort of high-density strontium and lithium bromide contrast agents, that came out in 1927. Incidentally, he's also the person who invented the lobotomy. He realized if you cut out fibers in people's foreheads, they act a lot differently. Sometimes it helps, maybe. So a lot of really bizarre, interesting practices that I'm sure wouldn't get FDA clearance today. But either way, then onward from that, you had William Oldendorf, who generated the concept for doing X-rays in a way that could give back projections and then allow sort of a 3D sort of X-ray image. And Hounsfield implemented that first and created the first CT scanner in the early 70s. And then now we have MRI. So that was all sort of coming about in the early 70s as well. And so, as you probably know, Peter Mansfield and Paul Lauterbur won the Nobel Prize in Medicine in 2003 for creating MRI. And what Lauterbur is known for is a couple of things. One, he came up with a sort of a rudimentary, sort of based on CT reconstruction, a back projection reconstruction using rotating gradients. He was able to reconstruct an image, the first images, using gradients of objects. So that was definite advancement. So Peter Mansfield actually came up with a concept sort of similar to what Lauterbur was doing, but using slice selection, actually came up with the concept of slice selection. So he invented slice selection. He also invented echoplanar imaging, which was, or came up with the concept for echoplanar imaging, which was theoretically possible then, but not physically possible then, just because they couldn't switch gradients that fast in human scanners. So had to wait until the mid-'80s for that, or the late '80s. But it's interesting, Raymond Damadian, who contributed to the field, he was a little bit of an outsider, he's an MD, he was kind of a maverick. He actually did also invent an aspect of NMR. But he just had a method for doing localization which was a lot more clunky and a lot slower, never caught on. The idea there was that instead of doing a gradient, he would have like a saddle magnetic field, and so it had one point that was resonant with the RF pulse. There was one point in the magnetic field that was resonant, and they would physically move this thing around to make the image. So it'd be physically moving around that sensitive point. So they made images. He made an image like this, but it was really kind of a clunky method that never caught on at all. So unfortunately, he didn't get the Nobel Prize. But there's still some debate as to whether he should have been included in that. So that's pretty much all I'm going to give really quickly about a rundown of some neuroimaging. If you wanted a little bit more background, I just took a sort of, this is a nice summary I got off the web of I didn't even talk about ultrasound, I didn't talk about optical imaging and things like that. But there's all kinds of things that have sort of came about in the mid to late, between the 50s and 2000 that are useful today. But I just want to jump into hemodynamic and metabolic imaging, though, as opposed to just purely anatomic imaging. So in some sense, the first experiment in hemodynamic imaging, and I don't even know whether it's really worked or not, but it's an interesting concept. And that was in the 1880s. This guy, Angelo Mosso, he put a person on a balancing beam, and they were delicately balanced. And the idea is that then he had the person either think really hard or give them some sort of stimulation or something like that without touching them. And the idea then is if there's more blood that rushes to the brain, then they'll tilt backwards on this thing. The blood, it's sort of a balance beam for blood. So, and he claimed it worked. And so that was the first idea, that blood rushes to the brain when it becomes more active. Along about 1890, there were some really, really beautiful experiments in animal models. This is in a dog brain. They opened up the brain, and they had these sophisticated devices that could actually measure cerebral pressure and brain position. And they gave the brain, the dog, stimulation, and they got what looked like a nice fMRI hemodynamic response with stimulation, looking at both the brain expansion and the blood pressure, blood pressure and brain expansion. And so that was in 1890. And that was pretty much one of the first measures of the hemodynamic response in that regard. And this is the journal article that actually inspired me. I was in high school when this came out, and I think I saw this when I was in grade school when this came out. I think I finally saw it by high school, and it was incredible. It just blew my mind that you could have, basically, the idea here is that you have subjects inhale xenon, which is a mildly radioactive substance. And then you have what's essentially scintillation counters that you put right at the surface of the head, and you measure where the radioactivity accumulates with activity. And so, they're able to act. This is Niels Lassen's group. He was able to map out, for the very first time, at least on one side of the brain on the surface, brain activity with, hearing words and seeing words and saying words and things like that. So that was just mind-blowing at the time. I thought that was the coolest thing. And then you had PET scanner that actually made use of the fact that when you have radioactive substances, they actually they have a annihilation sort of simultaneous radiation that goes both opposite directions. So the PET scanner actually uses scintillation counters, but also adds on the idea that there's a coincidence detector that allows you to triangulate a little bit better as to where the radioactivity came from to allow more in-depth maps of where the radiation deposited itself. So that was patented in 1973. It got better over the years. By 1995, it looked even better, and now we have simultaneous MRI PET scanners that give pretty high, maybe 3 to 5 millimeter resolution, I think, maybe higher than that even. So, and these are the images that came from there, which were also equally inspiring to me. This is a paper by Steve Peterson's group at the WashU in Nature, so from 1988. And okay, electrophysiologic imaging. So once again, back in the 19th century, 1875, you had the first currents measured between cortical surface and the skull in dogs and monkeys. And then you had the first EEG in humans, the description of alpha and beta waves in 1924 by Hans Berger. And about 50 years later, you had the first noisy measures of the magnetic brain signal, which is in 1968. But then something came along that actually allowed a much better detection, and that was in 1970 the SQUID devices, superconducting quantum interference devices, which really increased the sensitivity of mag. So allowed you to detect down to 100 femtotesla on the surface of the skull, or even less than that. So that helped things a lot. And by 1972, you had the first one sensor MEG recording with a SQUID, which was by David Cohen. So today we have 356 or 256 and more sensors, both MEG and EEG. You have whole brain EEG, and you can even combine EEG with MEG to get really both electrical and magnetic recordings. And you'll hear a lot more about this, of course, as the specific lectures. Rich Coppola will talk about MEG, and two other people will talk about EEG, at least 2 or 3 other people. So anyway, okay, so now into functional MRI. Okay, so this is something that was published in Neuroimage. So in Neuroimage, I in about a couple years ago, I came out with a special issue on when fMRI was 20 years old. And let's see here, what exactly was the date? 2012. So that's pretty much, fMRI came about in 1991, and so over the last 20 years, there were all kinds of pioneers who developed fMRI. And you could probably recognize some faces in this montage. And I would challenge anyone to try to identify all the names. Those are all the names right there. But so basically, there's 106 articles in this issue, and the reason why I mentioned it is because it really does. If you really want a good reference of the method development of fMRI, this is a nice reference because it's not necessarily technical papers. It's more papers by the people who did the development about their perspective of what they did and why they thought it was important and how exciting it was, or who are the competitors or whatever. And so everything from the first bold imaging to pulse sequences, to processing methods, to processing platforms, basic methodologic debates and things like that, novel paradigm designs, and then the future. We have a bunch of people who talked about the future as well. So anyway, that's sort of a quick plug for that because it's a nice reference. And I have copies of it in my office, so if you want to borrow copies or just download I have actually all the files digitally on my computer as well. I could package it up and distribute that too. So okay, so starting right now, as far as looking at the history of functional MRI, it's interesting because functional MRI it wasn't something like other methods that were like slowly developed, or it wasn't a gradual process. It was just something that was sort of discovered. And to give you an example, this was a paper that came out in Science, actually by some of the authors that were here at the NIH, Kurt Moonen, Peter Bandettini, Joe Frank, Denis Lebihan, and the only person I don't know is Edwin Becker. They were all at the NIH. And this was the state of the art of functional MRI back in 1990. And notice you had actually no functional MRI, what we actually call functional MRI. We have everything from angiography to gadolinium perfusion. You know, if you eject gadolinium, you get a map of perfusion. You have diffusion imaging. You have magnetization transfer, which was kind of hot back then, also developed by someone here at the NIH, Bob Balaban. Metabolic imaging was considered to be really exciting back then. It still is exciting, but it's sort of maybe lost a little steam because it's reached some technical limitations in sensitivity. But that was it. There was no hint of functional MRI. There was nothing. There was no statement, even in this paper, saying hemoglobin could be used as a contrast agent. In fact, there are many papers that said it couldn't be used and it wouldn't be useful. So that's where it was. And so it's all of a sudden in 1989 or 90, a lot came together. And so this is what I'd like to talk about. This is the 5 key factors for the emergence of functional MRI. So one, we had to be a little bit lucky in terms of the properties of red blood cells. 2, we had to be a little lucky in terms of how the brain causes blood flow and oxygenation to change. 3, we also had to get a little lucky in terms of the spatial scale of brain activation. 4, echoplanar imaging was critical, and it still is critical. And 5, the prevalence of MRI scanners really helped fMRI. So just to start out with red blood cells. So back in 1936, Linus Pauling was the first to discover that hemoglobin, when it was bound to oxygen, had a different magnetic property than deoxyhemoglobin. It turns out that deoxyhemoglobin is paramagnetic, and what that means is that if you place it in a magnetic field, it's not magnetic in itself. If you place it in a magnetic field, it concentrates the magnetic field, whereas oxygenated red blood cells are diamagnetic, which if you place them in a magnetic field, so the rest of the body is also diamagnetic. So they look like the rest of the body. So they're invisible when they're oxygenated. And that slightly distorts the field, but it makes it less concentrated. But since our whole bodies do it, it's something that it doesn't really change the magnetic field. So in 1982 then, Keith Thilborn, working with in spectroscopy, discovered that the T2 of blood was changed. And I'll get into this in a second. And then Ogawa also then made the discovery in 1990 that blood actually affected the contrasted images. And then Bob Turner also did the same thing in cats here at the NIH, looking at other things. So I'll jump through this here. So here's Linus Pauling right here, and he was basically he was starting out by saying that kind of reprimanding Michael Faraday. I mean, Michael Faraday, ahead of him, was initially characterized the magnetic properties of blood, but he always had it in more of a powder as opposed to in solution. So the paramagnetic nature of it apparently didn't come out that way. So he basically said, well, this would have been discovered a long time ago if the experiment would have done differently, and here I'm discovering that hemoglobin is paramagnetic. Okay, so Keith Thilborn was working with George Rada and at Oxford, and he basically found if he had a special pulse sequence that looked at relaxation rate, R2 is simply 1 over T2. It's the inverse of T2. And it turns out that there's a linear relationship between hemoglobin concentration and the relaxation rate, at least within that range that he studied. And importantly, he proved that it wasn't due to there's a number of mechanisms that can change T2. One is just intrinsic sort of dipolar interaction that happens at a very microscopic scale, and there's bulk susceptibility effects that cause dephasing of spins as they diffuse by these perturbers, these big perturbers. And so the idea then, if you change the spacing of the pulses that measure T2, you get a sense of, you change the amount of time the spins get to diffuse around these perturbers and become dephased. And if you do that, and if the T2 changes as a function of that shows that it's a bulk susceptibility effect as opposed to some intrinsic effect. So we proved that. And it was a really beautiful piece of work. So Ogawa then came along in the late 90s, and he's been studying, before this, he was studying blood for 20, 30 years using electron paramagnetic resonance, EPR. So he came along and working at Bell Labs, he discovered this, that if you have a rat at 7 Tesla with 100% breathing 100% oxygen, you get this image. And if you suddenly make it breathe 20% oxygen, you get this image. And so this is the idea that hemoglobin, when it becomes slightly less bound to oxygen, starts causing signal dephasing and decreasing the T2 such that you get this sort of image right here. And the idea here, once again, is this. If you were looking at a blood vessel, and this were the blood vessel in a voxel, and these are the magnetic fields, the microscopic magnetic fields that are set up around this blood vessel that's slightly paramagnetic. If you increase the amount of paramagnetism in the vessel, you'll get an increase in these gradients, which will cause, let's say, a spin that precesses, and all spins precess at the frequency in direct proportion to the magnetic field that they're experiencing. So these spins right here, at this edge right there, will be precessing much faster than these spins down here. So after a short amount of time, they'll become dephased. So the more steep these gradients become, the more dephasing you'll get, the more spin cancellation you'll get. So that's the basic mechanism that Ogawa outlined in 1990. Then Bob Turner in 1991, in the process of actually looking for diffusion changes in the brain, he found this. So here he made a movie of cat brain, a cat brain in which at this point in time, this is a series of difference images from this image. This point in time, they shut off the oxygen to the cat. And essentially, suddenly, within about, a minute or so, the whole brain starts getting darker. And that's due to the increased amount of deoxyhemoglobin that's in the blood because the metabolism keeps on going on. And it gets darker and darker, finally, differently in white and gray matter, proportional to the baseline blood volume. And then finally, he releases the cutoff, and blood rushes back, or oxygen rushes back in. Everything gets brighter. There's a reactive hyperemia here. And that was a really beautiful experiment that demonstrated whole brain modulations in oxygenation. Okay, and it's interesting then, from these experiments, Ogawa, it turns out, in his paper, I always like to quote this because, I mean, he was prescient in terms of predicting fMRI, but he actually, like most of us intuitively would, he got the sign wrong. So in his paper in MRM, he said, this might be able to show brain activation. However, with brain activation, you have increased metabolic rates, and it will pull more oxygen from the blood, the signal will go down. And so at least he got the idea right, that it could study potentially physiology, and he was actively working on it at the time. Turns out that in 1986, there was work that showed that in fact blood flow increases and oxygenation increases, and I'll get to that in a second. But back to this. So okay. So then from there, before Ogawa then showed bold effects in humans, before that happened, in November of 1991, Jack Beliveau, actually in August of 91, he was presented at a ISMRM meeting this wonderful new technique that looks at blood volume changes with activation. So essentially, he injected a bolus contrast agent, gadolinium. Within about a minute, it washed through the blood, washed through the brain, and during activation, during visual stimulation, you have a larger area under this curve that actually, he calculated the area under the curve, which is directly proportional to the blood volume, essentially. And he measured resting and activated blood volume. And two things happen with brain activation. One, the blood volume increases, and also the arrival time decreases. So the blood flow increases as well. And he came up with a map like this. That made the iconic cover of Science in November of 1991. But even by the time this was published, it was out of date because Ken Kwong was already working on bold contrast. And this is the group from MGH, which amazingly over the past, I would say 30 years has been innovating pretty much. And there's just a few people from the group, and you can see Ken Kwong, Van Medine, Jack, and Bruce Rosen right here. I think this picture was taken in Athens somewhere on some trip. Anyway, so now comes along. We're going to step back a second here to just show that in 1986, people, among them Peter Fox, were working on understanding neurovascular coupling. And in his 1986 paper, he realized, using simultaneous measures of blood volume and blood flow, that and also to get CMRO2 with PET, that it turns out that during brain activation, the oxygen extraction fraction goes down. So he realized that the flow goes up, the oxygenation in the blood causes the oxygenation to go up as well. So you have an overabundance of oxygenated blood going to the area. Even though the areas become metabolically active, they don't become active enough to pull the oxygenation down. The blood flow response dominates. So the area becomes more active. So this was already shown in 86. And so the basic idea here is that with cerebral tissue activation, you have locus vasodilatation, the increase in cerebral blood flow and volume that causes oxygen delivery to exceed the metabolic need. And so the reason why I highlight this is because this didn't really have to be. I mean, we're fortunate that this happened, that, it could have just matched the metabolic need. Somehow, consistently, the oxygen delivery exceeds the metabolic need. And we don't exactly know why that's important. And it seems like it's incredibly important because it happens all the time in a very consistent manner, but we don't really have a solid answer for why. And so then that causes an increase in cerebral capillary and venous oxygenation, and a decrease in deoxyhemoglobin then results, causing a decrease in the amount of paramagnetic substances there, which causes the signal to go up, the T2 and T2 star to increase. Okay, so here's Ken's first results. Ken was both looking at a novel arterial spin labeling technique that looked at blood flow, and here, it wasn't really looking at baseline blood flow, it was just looking at flow changes using inversion recovery, and this is his bold effect, and actually showing it agreed with PET in terms of the activation response. So it was a beautiful paper. Here's his notebook. What Bruce Rosen likes to say about this notebook is that that's his first experiment, May 9th, 1981, and he likes to show that it was original block design paradigm, and it used bold, it used inversion recovery. Okay, so other papers came out as well. Seiji Ogawa, collaborating with the Minnesota group. Turns out the Minnesota group was waiting on their high field scanner, a 4 Tesla scanner, before they even started their experiments, even though they knew that the effect should exist, and they had 1.5 Tesla scanners. They just were convinced that they could only see that 4 Tesla. So they finally got their 4 Tesla. They used a technique I'll get into in a second, but it was a multi-shot technique that allowed you to get a little bit higher resolution. And they actually showed that it was echo time dependent. So at a very short echo time, you hardly get any effect. At a longer echo time, you get this effect. And they also showed visual stimulation. And one thing I want to emphasize here is that really at the very beginnings, aside from the Minnesota group that did a wonderful job cleaning up their time series, the use of echoplanar imaging was really important. And I'll get into that a little bit later, but I just want to show, basically echoplanar stands for, when you give an RF pulse, you have T2 star decay of the signal, and you're ramping as fast as possible to get an image before the T2 star decays away. And there's something nice about this because this entire image is collected in 20 to 40 milliseconds, which freezes up all physiologic noise. You don't get artifacts that occur from different lines of K-space happening at different cardiac cycles and so on. So it's a super stable technique. Okay, so the first fast imaging approaches. So MGH, they had what's called an ANMR, Advanced NMR, was a company that retrofitted resonant gradient systems. That was on MGH. Minnesota had standard gradients, but with the multi-shot navigator echoes, with multi-shot, but with navigator echoes. And then MCW, the group that I came from, we had a local gradient coil for echoplanar imaging. So it turns out that before the MCW group could get any results, it turns out that Eric Wong was working on his thesis on gradient coil design, and he built this head gradient coil on a Next. He designed it on a Next computer. I don't know if you remember Next computers, but before Apple had its rebirth with Steve Jobs, Steve got it into the Next. But But he built this nice GUI that sort of used conjugate gradient descent to design these local head grading coils. This is Eric and I in August of 1991, sort of building it on a lathe out of sewer pipe, wire, and epoxy. And we needed a low inductance gradient coil so that the gradients could switch rapidly. With the standard gradient amplifiers that we had at the time, it would have just blown up the gradient amplifiers. So we just built that in about a weekend, and then we tested them out. This is me modeling our first gradient coil right here and a second one right here. Put in a lot of use for it. And the first experiments were, since we could only collect one slice, we took a 2.5 centimeter slice, just a big chunk of the motor cortex. And one little known fact, though, back then, is that we actually, even though we put all this effort into building this gradient coil, you didn't really need the gradient coil. Because if you just simply decided to image at 5 millimeter cubed on a standard gradient amplifier at the time, there were thousands of scanners with those standard amplifiers, you could run echoplanar imaging just fine at lower resolution, and you get perfectly good functional images at the time. You didn't need any special hardware. But anyway, we didn't know that when we built this and got a little bit higher resolution. And these were our first images back in 91, 92, right here. So this is then a movie of left and right finger tapping and so on. So this is right there. And this is my own notebook. One thing that I like to show in my own notebook is that we did it after Ken. We did it more in September. And what's interesting, though, is that definitely we got our initial input from the MGH group and the Minnesota group, but mostly the MGH group. They were a little bit ahead of Minnesota. One thing interesting, though, I was still thrown off a little bit by Ogawa's statement in his paper, and also at the August ISMRM meeting when they first showed the results. So Tom Brady was in charge of the NMR Center back then at MGH, and he showed these movies. He didn't say which way the signal went, so I kept on looking for signal decreases, and I kept on seeing signal increases, and it was confusing for a while, and that's what forced me to go back to the literature to figure that, a decrease in oxygen extraction fraction means the oxygenation goes up and the flow goes up. So now everything makes sense. Okay, so that was our contribution, at least back in 91, doing motor cortex activity in left and right finger tapping. What's little known, though, is that also there was another paper that was published right in the heels of this by Andrew Blameyer from Yale. And he pretty much invented event-related fMRI. And so he did basically 3-second stimuli and was basically showed event-related responses. So during this time, there were other groups. There was Alan Koretsky, who was at Pittsburgh, and John Detra, and Donald Williams, and John Lay, who were developing arterial spin labeling techniques. So these are methods for tagging blood inflowing into the image that would allow you to get non-invasively perfusion. And okay, so these are their first images right here. This is, like I said, came out in 1992. What's kind of interesting is that even though arterial spin labeling came out in 1992, what was very clear, and this is one of the unfortunate things about being part of the bigger MR scanner industry, is that while everyone had echoplanar imaging, and everyone had bold contrast, and everyone had all the imaging that we have, arterial spin labeling never came out as a product. And the reason why is because they patented it, and which is a good thing. You should always patent what you do, but none of the clinical vendors thought it was potentially useful enough clinically to buy the patent. At least it seems like that's the story. And so they waited exactly 17 years until the patent ran out, and now it's on all the scanners 17 years later. So that's one of the unfortunate things. If it's not a clinically useful or relevant sequence by the vendor's opinion, because fMRI is not a big clinical market, it may not appear. And so that's something we're always trying to fight. So anyway, the basic concept behind perfusion contrast is that it's like PET in a sense. You have a contrast agent, but the agent is not a substance that you inject. It's basically an RF pulse that you deliver below the head. So you give an RF pulse that changes the magnetization of the blood as it flows into the brain. And once it does that, the blood itself will change the magnetization of the tissue as it perfuses into the tissue. And that will affect the T1 of the tissue, longitudinal relaxation. So there's different strategies that came about. Epistar, invented by Edelman, who's more of a cardiology sort of person, but basically where you give a tag below and a tag above, and you subtract the pairs of images with FAIR imaging, flow alternating inversion recovery, Epistar's EPI, signal targeting, and alternating RF. With FAIR, you basically give a pulse everywhere, and then you then alternate that with the slice itself. And it turns out that if you can be very selective to different perfusion. So it turns out if you only wait 200 milliseconds, you get only the most rapidly flowing blood. If you wait over a second, you get more of the perfusing blood right here. And okay, so now let's switch gears a little bit in the early days. So this was one of the first conferences. It was in June of 1993, right here in Crystal City at the Ritz-Carlton Hotel. And it was it wasn't very big, but it was a very heady experience. A lot of people were very, very excited. You had all the names here: Denis Lebihan, Bob Turner, Michael Mosley, Jim Hyde. And it's interesting, there was even a lot of debate about what even to call fMRI. And for instance, Bob Turner was promoting the name MRFN. So he wanted to call it MRFN, which would be great because it makes you want to, it seems like it stands for more fun. So, and which it was. fMRI was much more fun than regular MRI. It was great. But that didn't catch on. So it turned into fMRI. By about 1993. So Bob Turner's contribution to fMRI, though, here at the NIH was that he showed that it was field dependent. So here at the NIH, they had the only 4 Tesla scanner, and so Bob Turner and Peter Jezard showed that the signal change doubled at higher field strengths. And so they used a local head grading coil that Bob Turner designed. Back in the old days, that's what he did. He designed grading coils, he made grading coils, and he made a Z coil just for the 4 Tesla. And this is Peter Jezard back when he was a postdoc for Bob Turner. And this is our 4 Tesla scanner, which was huge. Right now, this is sort of in the cardiac area. It's actually kind of around where the Prisma is right now. So, okay, and now, of course, you have Siemens selling their 7 Tesla, which is more of a turnkey system. Okay, so now with that, I just want to very quickly talk about the spatial scale of brain activation and why that was important. So it turns out it was fortunate, though, that the brain on some level is divided into large chunks. You have the behavioral control and cognitive areas in the front. You have motor cortex, visual cortex, auditory cortex, big chunks of cortex, at least at some organizational scale. And the same thing with the motor and sensory cortex. And that lent itself to fMRI because fMRI couldn't image at much higher resolution than 3 millimeter voxels, 3 or 4 millimeter voxels. So it was perfect that you could have a motor cortex activation and showing a big chunk of activation becoming active. It was, let's say the brain was divided, let's say this scale didn't exist. Let's say it's all layers, or maybe it's all columns. We would never be able to interpret these images with fMRI. Now fMRI is only starting to get into columnar and layer dependent imaging, but if this is how the brain was, fMRI would never have caught on. So that was fortunate that there are these big chunks of activation to show that fMRI works. But of course, the brain is organized across many different scales. There's the large systems level scale. There is organization across layers. There's organization within columns. And it turns out the whole brain might end up being completely, the most interesting organizational structure might be layer and columnar organization, even in the frontal cortex and everywhere else. That's yet to be found. So also in the early days of fMRI, some early positive results with showing columnar activation, ocular dominance columns that matched these optical imaging results by Ron Frostig pretty nicely. This is some high resolution work by Kang Cheng. And this is some more recent work by Essa Yacoub at the Minnesota group at 7 Tesla with human V1, showing orientation columns. So each one of these color codes is encoding a pinwheel as a grating is being rotated. So you basically show activation, and the color phase encode is a sort of a phase encoding of that grating. So you can get down to orientation columns, which are relative to ocular dominance columns. Each one of these is an ocular dominance column. And so even here at the NIH, in our group, Laurentius Huber has shown layer-dependent activity. So here he has a very specific type of finger tapping task, looking at finger tapping versus not finger tapping. And the idea there is that you have sensory input into the motor cortex that's modulated by either the sensation of tapping versus not tapping. And he was able to show specific layers, layer 2 and 3, showing more activation with the tapping task than without the tapping, just movement without tapping. So that's the upper limit of resolution. And like I mentioned, it could be everywhere. So this is some work by Mike Beauchamp showing that even in the multisensory integration areas, more higher cognitive areas, in the parietal area, lobes, you have this interdigitated activation between visual, auditory, and multisensory activation. It's no longer these large blobs. It turns out the higher resolution you go, the more interdigitated it becomes, which becomes more interesting and more of a problem for processing. So this pretty much summarizes a lot of the various techniques. You've probably seen this diagram many different times, so I don't really have to go into it. There's always debate as to how to draw this MRI curve just right. Is it the actual resolution of MRI, or is it the functional resolution? I don't really care too much. I mean, I do care very much about pushing this envelope, but most importantly for why fMRI took off is because it was this massive chunk right here that was clearly higher resolution than PET, a lot easier than PET, not as invasive as PET as well. That was also higher resolution spatially than MEG and ERP. That was non-invasive. So all these areas of the brains are those large chunks of activation that lend themselves to saying, oh, well, this is the attention network or whatever that shows modulation with activation. So we couldn't even do those studies without using MRI, something that's non-invasive, you can do over and over again, and it just hits that sweet spot of systems-level organization of the brain. Okay, now to just jump quickly into echoplanar imaging. As I mentioned before, it means one echo. For each echo, you get a plane of information, and essentially, you need high-performance gradients to ramp back and forth in K-space very quickly. The contrast, as opposed to MRI, anatomic MRI, is not over space. These are really horrible images. I should update these. Because this is what the first echoplanar images look like. I mean, some of the first ones that didn't have the best phase correction and were very low resolution. But the basic concept here is that you're looking at the contrast as it evolves over time and not over space. You're only comparing over time. And once again, just to reemphasize the difference between echoplanar and multi-shot techniques. And this is something that's often missed in many different types of studies, and that is that, when I talk to people about the difference between multi-echo or multi-shot versus echoplanar. So with echoplanar, you might have ghosting. You certainly might have ghosting, but the idea is that because all of this is collected within 30 milliseconds, the ghosting will be identical with every single image because physiologic noise is pretty much frozen in that 30 milliseconds. But with multi-shot, this takes anywhere from, well, let's say it even takes 2 seconds. So 2 seconds to 1 minute to collect. You might have phase shifts in your raw data due to the fact that for instance, when the heart beats or when you breathe, that does cause phase shifts in the brain. And that causes misalignment of the K-space lines. And then when you do a Fourier transform of this, you'll have ghosts as well, which is like you would have with echoplanar. But the big problem here is that you never capture the heart rate and respiration rate the same way for each successive image. So the ghosting is non-repeatable, and that adds a ton of instability to your time series. So that's why echoplanar was so important for doing functional MRI, because it really, really stabilized the time series. If everyone tried to do flash imaging over time. It would be slower, but also it would be a lot noisier, and it would be much more difficult. So looking at the echoplanar imaging timeline, I'm not going to go through this in great detail, but just to try to emphasize that by about, there are a couple of things that helped a lot. Of course, echoplanar, parallel imaging allowed for higher resolution EPI. Oh yeah, by the way, 1996 to 98, it was standard on most scanners. By 2006, single shot EPI, 1.5 cubed resolution was possible. 2009, at 70 sub-millimeter single shot EPI was possible. And I didn't even really get into multi-band imaging, which came about 2010 or 11, so which really picked up after that. Okay, looking quickly at imaging system components, you have the gradient amplifier power system, which now has improved a lot. So that allows us to not need head gradient coils. We can have the whole body coil and do just fine. Okay, so the last part before I end is just to talk a little bit about the prevalence of MR scanners and how this helped fMRI. So this is what I like to say here is that, well, one theory is that this is basically the progression of papers up to about 2008 of functional MRI. Functional MRI was a technique that worked really well from the start, so it would have caught on regardless. Up until 1996, though, all the systems that were doing echoplanar were all home-built, or sort of these one-off companies. And so the progression of the number of papers was pretty low. It probably would have just kind of been like this, I think. But suddenly, echoplanar imaging became available on all scanners, and pretty quickly this curve changed slope and it took off. That's my theory. And another thing to emphasize is that we really are riding pretty much on the back of the clinical imaging industry. So typically what happens in most brain imaging techniques, let's say you have EEG, for instance. Well, not even EEG. I can't even think of any, like ultrasound, for instance, or some other method that MEG is a good example. Okay, so MEG is not that popular clinically. There's a few niche applications, but it really is not that popular. And so but in research, it's much more popular. You have a lot of groups doing MEG. The problem is, though, is that there's just not. They're still very expensive. There's only a few companies that are only selling a few scanners, and it's sort of an anemic industry, and it hasn't really propagated that much. Whereas with functional MRI, so all of a sudden, once fMRI was discovered, and especially once echoplanar became available on all these clinical scanners, suddenly every single clinical scanner in the world was a potential fMRI machine, which was incredibly helpful for the propagation of the field. So that was extremely important. And so we're both very lucky to have the clinical industry, but at the same time, we're always struggling with trying to get our foot in the door a little bit more and saying, hey, fMRI is important too. Could you please help us a little bit more? So that's pretty much summary of my talk. I just wanted to also bring out the fact that it's gone way beyond just finger tapping and visual stimulation. And this is a nice slide that I still use, even though it's very old, from Judy Isles, that just kind of shows how fMRI developed. Very quickly. By about 1997, we became so confident in the signal and its reproducibility, and it applies across all parts of the brain, that suddenly, it was being used for all these different things and barely for visual or motor cortex activation. So even now, like in NIMH, we have entire programs devoted, that are based on using fMRI for their studies. So that would be unheard of 20 years ago. So, and then another thing I'd just like to emphasize just before I end is that the field itself is, I like to look at it as having these four pillars of advancement. There's the basic technology, which is, it's constantly being pushed, the coil arrays, high field strength, high resolution novel sequences. You'll hear all about that in all the lectures coming up. You have the methodology, the processing methods, things like that. And you have the interpretation of the signal, getting a better understanding of what really the signal means, because we're still trying to figure that out. And And then finally, the applications that sort of draw from all of those and also inspire them as well. And I like to think that some of the best work is being done right at this interface. So that, in my opinion, the field gets pushed by, of course, pushing any one of these, but the really coolest work is usually right here that makes, uses the latest in the methodology, the latest in the technology, uses some aspect of our understanding of the signal that lends itself to the question being asked and addresses it right here. So that's continues to go, and these areas continue to interact. And so I'd recommend, as you do your research, to try to learn as much as possible about all the aspects of fMRI, because it really is valuable, because the field is really still advancing rapidly. And to understand all the moving parts, I think allows one to sort of kind of be on top of things better. Okay, with that, and also this slide, of course, is going to be available online within a week or so. But I just added a bunch of old papers that might be good landmark papers to take a look at for your reference. So yeah, with that, just like that. Thank you. Thanks. So one thing I forgot to mention is that certainly you can interrupt me or any of the lecturers during this course and ask questions. It's totally informal. It's nothing. Yeah, I'm happy to, and also I'm happy to answer questions now, if you have any questions. So, yeah.
So maybe I'm misremembering things, but I started off grad school around this time when fMRI started to take off a little bit. And my recollection was that at the very beginning, there were still a lot of people that didn't believe that fMRI was actually measuring something reasonable. So can you talk about maybe when the turning point was, where it really became more accepted? Or am I misremembering things?
Yeah, what was the guy's name? Let me think about this for a second here. I forgot the guy's name. He's still in the field today. There was a couple of people who thought it was all motion. There's some people who thought it was all, there's a lot of debates. There's some people who thought it was all large vessels, who thought, oh, this is just large vessel effects. And there are some people who actually they were attempting to do multi-shot imaging at very high resolution, and they couldn't see anything, and therefore they said, oh, this I can't see anything. And that was a problem. And so what happened was, everyone, they would go to meetings and they would get a lot of attention, like, oh, this isn't real. And I think pretty quickly what happened, though, is that I think that there are two experiments. One, early on, there was that beautiful retinotopy experiment by Marty Serino that I think helped put the nail in the coffin of any doubts that it was real. Because there's no way you can, if you do retinotopy, you give a visual stimulus and actually show an expanding ring and then show the expanding activation, there's no way you can sort of create that by moving a certain way. And so that helped convince the field that it was real. There are a few other experiments like that. I think just what happens in science is that, quickly, yes, you have doubters, but people just keep on turning away results, and you just get overwhelmed by the, enough people were producing successful results rapid enough. So, but I really do think that retinotopy experiment was a key experiment. That was like in the mid to like 1996 or so. So between 92 and 96, there was still some debate about fMRI and that sort of thing. But yeah, but quickly it vanished. So yeah. Okay, great. All right, well, I'll see you on Monday then. All right, thanks.